Hi, and welcome to our course on protecting future generations. We're going to be talking about the science of epigenetics and how that relates to living a chemical-free lifestyle with you and your pet. So welcome, and I hope you enjoy this class. So let's start out by talking about our audience. Some of you are dog breeders and some of you are pet owners. And as such, we're all invested in the health and wellness of those that we love. As breeders, we are stewards of our breeds, and it's our responsibility to protect them from genetic diseases and from chronic illness. And the purpose of this course is to give us practical and simple ways to apply genetic science to everyday life with the goal of protecting future generations from the harm that's unintentionally inflicted by the environment. And if you're not a breeder, you're going to benefit by understanding how this can protect your animal and yourself in everyday life and help your pets to live the most vital and long life possible. When I went to college to study to be a veterinarian, we were taught that genes were like this kind of you get what you get and you don't throw a fit. You know, if our parents had diabetes, we were very likely to get diabetes too. And that was absolutely correct. But what we weren't taught was that we could change those genes during our lifetime and affect future generations by doing so. Epigenetic science is proving what I was taught to be incomplete at best. But most definitely it isn't new. The term epigenetics, which was coined by Waddington in 1942, was derived from the Greek word epigenesis, which originally described the influence of genetic processes on development. During the 1990s, there became a renewed interest in genetic assimilation. This led to elucidation of the molecular basis of Conrad Waddington's observations, in which environmental stress caused genetic assimilation of certain phenotypic characteristics in Drosophila fruit flies. Since then, research efforts have been focused on unraveling the epigenetic mechanisms related to these types of changes. Epigenetics is even alluded to in biblical texts. You know, the comments, the sins of the father being expressed upon his children for 3 to 4 generations. The science of epigenetics is now revealing that epigenetic damage actually can last for 3 to 4 generations before it can be fully reversed by converse influences. So what are those influences? And what did they mean by that in the various revelations of the Bible? In order to believe in epigenetic influences, one might want to know a bit of the science. So here goes. Really, stay with me. This is short. Everyone has seen the double helix of DNA, right? This double helix contains the instructions that tell the cells how to behave, what to make, and how to protect the body. DNA contains instructions much like writing on a piece of paper. When that paper is open, the instructions are easily read. However, if environmental triggers change epigenetic markers, that paper containing the instructions basically crumples up and cannot be read properly anymore. You might be able to read part of it sometimes, but not all of it. You can think of these epigenetic changes as ones which can crumple or uncrumple the paper containing the instructions for good health. But what you may not know is that the strands of DNA are wound up very tightly by proteins that are called histones. Epigenetic changes alter the physical structure of DNA. One example of an epigenetic change is called DNA methylation, the addition of a methyl group or a chemical cap to part of the DNA molecule which prevents certain genes from being expressed. Remember the word methylation because it becomes important soon. These proteins, called histones, are wound up with DNA, affecting whether they are able to express. Just like when you wear a tight dress and can't breathe, tightly wound DNA cannot breathe and is unable to actually express itself. Methylation can activate or repress genes by affecting how the histones wind or unwind. Histones wrapped tightly can stop a gene from expressing. Let's say the gene for degenerative myelopathy in boxers, for example, or the gene that you may have inherited from your mom that makes you prone to getting migraine headaches. Histones, unwrapped, can allow that gene to be activated. So in this slide, I want you to come away with the understanding that we need methylation, and there's an excellent source for it from various places, including Young Living, that can be added to diet. Another example is histone modification. Histones are proteins that DNA wraps around. Without histones, DNA would be too long to fit inside cells, since each cell contains about 6 feet of DNA, about 400,000 times the length of the cell itself. If histones squeeze DNA tightly, the DNA cannot be read by the cell. Modifications that relax the histones can make the DNA accessible to proteins that read genes. Epigenetics is the reason why a brain cell, for example, that comes from a genetically identical stem cell doesn't look like a muscle cell that is genetically identical. All these cells contain the same DNA, right? But their genes are expressed differently. They're turned on or off, which creates the different cell types. Epigenetics literally means above or on top of genetics. It refers to external modifications to DNA that turn genes on or off. These modifications do not change the DNA sequence, but instead they affect how cells read the genes. So when you look at something like a caterpillar that goes into a metamorphosis and becomes a butterfly, it has the same genes throughout the whole process of its different life stages. So the question is, how do we modify these epigenes in either a positive or negative way? Mother Nature influences epigenes by affecting their expression with natural chemicals. So that's how a caterpillar with the exact same genes becomes a butterfly. It's a chemical process. We can't make a butterfly, but we can make a difference. In this slide, we have two guys that have the same genetics. The guy on the left eats healthy, he exercises, he gets fresh air, he gets sunshine, he's mindful of his environment, he reads labels, and he learns how to identify potentially hazardous chemicals in his home. The guy on the right hangs out in the dark. He eats a lot of processed foods, he cleans with the cheapest thing he can find on sale at the big box stores in bulk, if he actually cleans at all. He washes his hair with shampoo containing sodium lauryl sulfate, parabens, triclosans. He watches disturbing news 24/7, which keeps him pretty stressed. And each of these men, if they reproduce, has the potential to pass their phenotype, which is how they express their genes, along to their offspring. Phenotype is the expression of genes, while genotype is the genes themselves. These guys have the same genes, but they pass along different characteristics to their kids. Genes provide the instructions for the body, whereas epigenes decide whether those instructions are actually carried out. This may be why we see dogs genetically screened as, for example, at risk for a certain inherited disease like degenerative myelopathy, never actually express the disease. We know that this is true in about 90% of the dogs, for example, in my breed, boxers, who are genetically at risk for degenerative myelopathy. A study was conducted on genetically identical rabbits. Some rabbits were exposed to high temperatures above 25 degrees centigrade, and others, genetically identical, were exposed to much colder temperatures. The rabbits exposed to higher temperatures expressed white color. That was their phenotype. Rabbits exposed to lower temperatures expressed black coloring on their noses, ears, feet, and tails. What's interesting is that even though their basic genetics were identical, these epigenetic traits passed on to future generations. Interesting, huh? Here's a story of two mice who are genetically identical. These are actually genetically identical twins. These are both agouti mice. They're originally bred to be yellow, obese, and disease-prone. They're used in scientific studies. The difference between these two mice is simply that the normal, healthy brown mouse on the right had an epigenetic influence. His mother was fed a better diet with proper supplementation. The little guy on the left was raised by what I like to call the meth house mice mom, a mom exposed to poor diet, and thus her environment was affected. Genetically identical, but profoundly different animals. And these traits have been shown in animals to continue for tens of generations in mice. In humans, the science is still debated, but the animal science is well proven in mice, in worms, and flies. Epigenetic markers are passed on to future generations, and they can be good ones or they can be bad ones. The current sciences of epigenetics is really interesting. Drugs have been developed and are FDA approved and in clinical trials right now to actually turn off epigenetic markers that wrap around and silence genes whose job it is to suppress tumor formation, for example. This is a huge departure from the traditional thought that cancer cells must be killed. Instead, we're giving the cells a little reminder, if you will, to turn back on and start suppressing cancer as they are originally designed and programmed to do. We know that certain diseases are also affected by aberrant genetic markers besides cancer, like diabetes, lupus, asthma. Certain neurological disorders are being studied, such as Huntington's and Alzheimer's, that may be affected by these epigenetic markers. Scientists are studying ways to target the markers that affect negative disease states while leaving alone the markers that affect positive healthy states. That's still underway in medical science. Research and trials are in full swing in many places in the world, and even in my own backyard of Houston, Texas. MD Anderson, Baylor College of Medicine, and Methodist Research Institute, to name just a few, have these studies underway. Studies are being conducted on DNA methylation and histone aging as well. This is still a relatively young science, but there are things we can do now to positively affect our epigenome. It's not too late to start eating healthier foods and feeding our carnivores a high-quality raw meat diet as they are designed to eat. We can reduce stress. We can be mindful of known toxins in the environment. We can provide lots of fresh air and sunshine. This is a profoundly different way of thinking to affect disease states. We always thought that our genes were set in stone and beyond our influence, but this science is proving that this is not the case. As breeders and as pet owners, what this means to us is that we need to understand a little bit about these epigenetic influences in the environment. We need to be feeding appropriate, high-quality raw meat diets to our carnivores, to our cats and dogs. Cats are obligate carnivores, which means that they are obliged to eat raw meat. Dogs can kind of do okay. They can survive on a kibble diet, but they don't thrive. We need to be adding antioxidants, trace minerals, and whole food supplements if that's appropriate to complete those diets. We need to be using natural heartworm and parasite deterrents instead of the systemic insecticides that are so commonly used in the veterinary practices. There are natural antibiotics and essential oils that are available to support body systems instead of using synthetic antibiotics that are responsible for bacteria that are resistant to antibiotics such as MRSA. We can also be using homeopathics and flower essences with our pets and with ourselves. And it's certainly important to get the toxic household products and cleaners out of our pets' environment. We also need to reduce vaccination with heavy metals. They have foreign proteins from other animal species in them as well, and they do have an epigenetic influence on our animals. What's not on this slide too is that we need to be giving our animals healthy ways to reduce stress. We need to be engaging their brains and bodies in healthy ways. We may be affecting future generations in incredibly positive ways by being mindful of these epigenetic influences. We may be able to increase vitality and longevity and pass those traits on to future generations. I think this is a very cool concept, and I personally see no downside to embracing such a lifestyle for our animals and for ourselves. When we talk about household chemicals, there are some pretty common ones that are very harsh and quite toxic that are still used in the United States and often banned in other countries. Products that contain BPA, triclosans, parabens, sodium lauryl sulfate, and phthalates are always on the top of my list. These products are fairly easy to identify by typing them into the website that is called www.ewg.org, which stands for Environmental Working Group. You can also use an app for iOS and Android that's called Think Dirty, and you can scan barcodes to see if a product that you're using or thinking about using is on their hit list. This list that's on this slide is produced by the Environmental Working Group, and it's called their Dirty Dozen. Those are things that you might want to look for on labels when you are deciding what products are safe to bring into your home. One of the easiest things you can do is to identify and replace these harsh chemicals in the home. Pets are low to the floor, and they have much smaller body mass than humans. They often lick chemicals from their fur and their feathers. Breeding programs are expensive, I know. We all know that. We all need to conserve funds. And I've found ways to do that and still maintain a very high level of environmental cleanliness. When I learned to read labels mindfully, I discovered that many of the household cleaners and self-care products that I was using are on the Environmental Working Group's list of dangerous products. In general, the most egregious categories include synthetic air fresheners, many household cleaners, products that are used to deter insects or kill parasites, laundry detergents, dryer sheets, kitty litter, antibacterial soaps, and even tap water. Products containing triclosans, parabens, SLS, and phthalates are always on the top of my list. These products are fairly easy to identify by typing them into the websites that I mentioned, and you can replace those products with a variety of natural, safer choices. It's not my intention to sell anything in this class. If you want to purchase any of the products that we use ourselves and recommend to our students, you can talk with us or get with a Young Living member who invited you to this class. We do run a large team of passionate Young Living members, and we're always happy to sponsor and teach people into Young Living so that they can replace potentially hazardous products with ones that may be more epigenetically conscious. There are practical things that we can all do to protect ourselves and future generations. This is our passion and our purpose in life, to help our fellow Earth citizens to stay well and to protect their children and pets. This is a picture of Ginger. our veteran boxer who has walked this path with us, teaching us about the horrors of turning on negative epigenes and the joy of reversing them. She was 5 years old in the picture on the left. She was vaccinated for the third time at age of 3, and this pushed her into a severe rabies miasm. Her thyroid numbers, they went from really very nice, beautifully normal, to off the charts with autoimmune thyroiditis. That means that her body saw her thyroid as a foreigner and spent a lot of its own energy trying to get rid of it. She really became an old dog at the age of 3. She developed arthritis, and she began to fray one of her cruciate ligaments just from normal daily walking activities. She looked like she was 100. She could barely get up in the morning. And after a long process of discovery and understanding of the dark side of heavy metal poisoning from vaccines, we worked with an enlightened veterinarian who suggested some natural supports that helped her to bring her own body to a state of homeostasis. She went from an arthritic old lady to a vibrant senior who runs athletic events at high speed. Her joints no longer hurt, and she never required surgery to fix her cruciate ligaments. She was the oldest boxer entered in the 2017 American Boxer Club Nationals in her class, where she wowed crowds with her vigor and vitality. She was the third oldest boxer present. She's a beautiful example of the profound changes that can be had when one is mindful of epigenetic influences in the environment. She will forever need thyroid supplementation and support, but she has the body and the mind of a youngster at the age of 10. The average lifespan of a boxer is currently 10 and a half, so we're looking forward to many more years of this young lady living with us. I want to end today by making this challenge to you to take this information and implement it in the care of your pets and in your breeding programs, to influence long-term health in positive ways by treating our epigenome kindly, by feeding our pets a healthy, fresh meat diet with proper supplementation and including antioxidants and MSM for methylation, by eliminating as many known toxins as possible from your pet's environment. Consider all aspects of the medicine you may have in your arsenal and think twice about using them just because you can. I was taught by mentors whom I respected to use many things like synthetic antibiotics prophylactically just in case. They actually do a lot of harm long term. They're used during pregnancy a lot of times just to ward off anything that might happen. It's the most dangerous time to compromise a mother's immune system, and using synthetic antibiotics during pregnancy does affect a mother's immune system in negative ways, and it does affect her epigenes. As you can see, this may affect their unborn puppies and the generations that follow. Be mindful of systemic insecticides as well, the ones that are given topically or orally, as many of them are known to provoke neurological damage and even death. Take it upon yourself as custodians of those who cannot choose for themselves to provide a quality diet, fresh, unfluoridated, and not polluted water, and an environment as free of harmful chemicals as you can possibly afford. I believe that we can improve our breeding programs through these mindful choices, and we can bring these beautiful, funny companions into their wisdom years without the chains of crippling arthritis, degenerative myelopathy, heart disease, and the many other health issues that plague our seniors and even our youngsters. This is a picture of Baron. He's a boxer who lived to be 19 and was chasing frisbees until he went over the Rainbow Bridge. That's around 115 for a human. This dog was raised traditionally in an environment without much in the way of chemicals, and he had loads of sunshine and fresh air, access to plants and forests, and love. He was not a kennel dog. If Baron can live to be 19 with a diet full of carbohydrates, kibble, and preservatives, think what might be possible with a sound epigenetic basis established for our puppies ahead of time.